Hello, hello, hello. Welcome to Genetics Day 7, and we are about to talk about gel electrophoresis, baby. So let's go ahead and get started. All right, so today's aim is how can we analyze DNA? And analyze really just means obviously to look at closely. And what we specifically mean in this case is we're going to analyze DNA to see which DNA is similar to other DNA. So start thinking about why might we want to compare DNA? Why might we want to see if one strand of DNA is similar to another strand of DNA? Start thinking about that. And our first question here, the do now, says the following. Experiments revealed the following information about a certain molecule. It can be broken down into amino acids, so that tells us something, right? This must be a protein because amino acids are, of course, the building blocks of proteins. Then it also tells us that it can break down organic molecules into their subunits. So it can break down. Another word maybe for breakdown is digest. Sounds like something we've talked about before. And its function can be severely reduced if it is exposed to high or low pH conditions. So it operates in an optimal pH range. Does this sound like something we've heard about before? Answer it on your own if you can. Pause this video. Otherwise, this molecule is most likely an enzyme, right? Enzymes are proteins that can break down organic molecules into their subunits, right? Enzymes are involved in digestion. And this question then asks us to define enzyme. So you might have to think back to Unit 2, Unit 3. How do we define an enzyme? An enzyme is a biological catalyst, right? It's an organic compound, a biological catalyst that controls the rate of chemical reactions. And we actually use enzymes to help us analyze DNA. So let's talk about it. First, before we talk about it, remember I told you to start thinking about why might scientists want to compare DNA? Why might scientists want to compare the sequences of DNA between two people, for example, or two organisms? So go ahead and pause this video and write down why you think scientists might want to compare DNA sequences between two people. So some reasons scientists might want to compare DNA between two people to see how similar the DNA is, which we might do in a paternity test, right? So to see who is the father of a child, right? Like think about Mari. You are the father, you are not the father. I'm sure we've seen the memes. Or to determine familial relationships or ancestry, right? Like Ancestry DNA or 23andMe that you may have seen advertised online to determine family DNA, family relationships, or ancestry. And another one that you may have been thinking is to solve a crime. So we have a DNA sample at the scene of a crime, and we're going to compare it to the suspect to see if they are the one who committed the crime, right? Which is also why you never give the police your DNA. Not saying that you committed a crime, but just don't ever give the police your DNA. Just don't ever talk to the police or answer their questions. Just a good general rule. But something else, some other reason that scientists might want to compare the sequences of DNA between two organisms is to examine evolutionary relationships between species. So if two different species have similar DNA, it might indicate that they evolved from a common ancestor. And we're going to look at that in a lab in our next unit. So you probably didn't come up with that one on your own, but go ahead and write it down. And I know yesterday's video was a long one, so I'm going to try to keep this one a little shorter. Let's see if I can do it. So we're talking about gel electrophoresis here. So this is a key technique for analyzing DNA. And this is really the only one that we're going to talk about. So we can see over here, this is an example of how gel electrophoresis happens. So we can see there is a sample, a DNA sample that's being put into a well. These are the wells where the samples are placed. So we might have sample A, B, C, D, E, F, and G, and we're going to be comparing them to each other. So we have the side where the DNA is placed. Up here, this has a negative charge, and DNA is naturally negatively charged. So DNA is negative, and a negative, when it is added to electricity, is going to be attracted to a positive. So the DNA is going to travel in this direction towards the positive end. So an important element here, something that is sort of not pictured, but this is called gel electrophoresis. So this cathode and anode, this is charged with electricity. Right, so the electricity is very important. And then we can see there in the gel, those little lines, those are the DNA fragments. But how exactly does that happen? Well, gel electrophoresis, to put it simply, is a procedure which separates DNA fragments according to their size, using an electrically charged gel. So the DNA samples are placed on the negative end in the sample wells, and then they travel from negative to positive. And we're going to talk about exactly how they travel here. So first, before the DNA samples are placed in the wells, in the procedure, this is why we started talking about enzymes, the DNA samples are cut into fragments or pieces using a restriction enzyme. So we can think of a restriction enzyme is like molecular scissors, right? So I'm going to draw a little scissor here, although shout out to Tiffany in period 7 who told me my little scissors looks like a butterfly. But we use this to represent the restriction enzyme. molecular scissors that cut the DNA. So these enzymes, we're going to define them here below. Enzymes recognize and cut specific sequences in DNA. What do I mean by that? So let's say, for example, specific, for example, they cut between C, C, G, G. So every time in the DNA sequence, there are two C's followed by two G's. The restriction enzyme will cut, but not at other points in the DNA. So these restriction enzymes have specific action. Then the next step in gel electrophoresis, we're going through the procedure here, is these DNA fragments are pipetted into a special sponge-like gel. So we're going to zoom in on this image over here and take a look at it. So first, the DNA samples are cut with the restriction enzyme, and then they are loaded into wells. And we can see here, the end where they are loaded in is negatively charged, and the DNA samples are negatively charged. So they are loaded in here with the pipette. And let's say we have sample A, and this is the baby. And sample B, C, and D are the potential fathers. Right, so we don't know who the father is. This is an episode of Maury. Mom was really busy, and we don't know who the father is. So how are we going to find out? Well, by using this process of gel electrophoresis. So the DNA samples are first loaded into the wells, and then the power is turned on because this process is electrophoresis, right? We need power. And the DNA fragments then migrate through the gel. So they're going from negative to positive, and the largest bands will wind up closest to the negative side. So larger bands don't move as much, and they also move slower. So the charge is not able to move them as much because they're larger. And they're going slower. So taking a look at this image, you can see down here below, the smallest bands are over here closer to the positive side. So this is the smallest DNA fragment. And looking up here, this is the largest. And by comparing sample A to B, C, and D, we're going to see it looks like, and we're just looking at the lines, how they read out, right? So each line represents where the fragment of DNA stopped, where the restriction enzyme actually cut that fragment of DNA. Based upon this readout in this diagram, we didn't find the potential father, right? So you can see, well, A is the DNA of the baby. There's one commonality here with B. There's one commonality there with C, and there's no commonalities there with D, which means we did not find the father. But we're going to get a little practice with that below. So that was step 2, but we talked about really the whole process. Step 3 is electricity is applied to the gel. This is gel electrophoresis after all. That is a little lightning bolt. This is electric, boogie, woogie, woogie, making one side of the gel negative and the other side positive. And the DNA is going to move from negative to positive charge, which means it's pulled towards the positive end because opposites attract, baby. And that's also, like how electricity works. The fragments that travel the farthest are the smallest and they move faster. So they'll get all the way to the end first, and then the ones closer to the negative end will fill in. Unfortunately, we're not actually going to be able to run any gel electrophoresis tests live, but if you watched one happen in the lab, that's what you would see. And the DNA fragments, as the DNA fragments get trapped in the gel, they form visible bands. And that's what we're seeing here in this diagram. These are the bands. So when we compare, we say it is the banding pattern. And with this little bullet here, you can leave it blank, or you can remind yourself the DNA travels from negative to positive. If the DNA fragments from 2 samples settle in the same place, it indicates a relationship. And the way we say that is a similar banding pattern. So congratulations, you are the father. So now let's do a little practice. First, we're going to use a restriction enzyme. So step 1, the restriction enzyme HAE3 cuts DNA sequences between the bases of G and C. Indicate where the enzyme would cut the DNA sequence below by inserting a line. Afterwards, determine the number of DNA fragments that would be produced. So we're going to cut all the way through based on the first line, okay? So take a look. And we're looking for G, followed by C, GC, GC. So you can see there's CG, but we're going based on the first line here, and we're cutting this into 3 fragments. Count them, 1, 2, 3. So 1 short fragment, 1 much longer fragment, and a 3rd small fragment at the end. And remember, fragments just means pieces. So step 2, after digesting the DNA molecule with HAE3 in step 1, how many bands would you see if you ran the fragments through an electrophoresis gel? Why? So we would see 3 fragments or bands in the gel. based upon our results in step 1. Or how the enzyme cut between C and G. Or rather G and C, excuse me. All right, so question number 2. If DNA fragments from 2 samples settle in the same place, then the segments of DNA are common. So that means they are a match. And we're going to get a lot of practice with this during class. So if you're still a little confused at the end of this assignment, don't worry, we're going to practice with gel electrophoresis during class. But let's take a look at this one, number 2. So the diagram on the left illustrates a gel electrophoresis test comparing DNA fragments of a baby's sample to 3 possible fathers. Based upon the results of this test, which man is the baby's father? Explain how you would know. So we can see the baby here, and we can see 1, 2, 3, 4, 5 DNA fragments. And based upon where the DNA fragments land, we're going to be able to tell a relationship. So let's go ahead and see. We can see one commonality with all 3. Uh-oh. 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 So it looks like if you were able to follow as we were going through, we were just looking across and seeing where the bands matched up. And in order to be the father, it doesn't need to be exactly the same, but in this case, we can see the baby and man number 3 have identical DNA banding patterns. So man number 3 is the father because when comparing the gel electrophoresis results, they had the identical banding patterns, meaning they had the same, congratulations, the same DNA. Congratulations, he is the father. So go ahead and complete your exit ticket. Today's aim was how can we analyze DNA, and we examined the process of gel electrophoresis, which starts with a restriction enzyme and ends with looking at the DNA banding patterns. So I hope you learned something there. Have a super fantastic day, and I'll see you guys all tomorrow on Zoom. Bye, guys.